Next presentation is our second interdisciplinary team with biomedical.
Great. Well done.
This one is team number 3, integrate and image.
Hi, I'm Craig Laboda. This is Austin Collins, Nika Galili, and Shannon Grover, and we are Team 3, Integrate and Image. We are also an interdisciplinary team with Austin and I as the EE team members, and Shannon and Nika as the BME team members. Our advisors for this project are Professor Unlu, Professor Kabodi, and Professor Clapridge. And today we'd like to talk to you about our integrated device for infectious disease diagnosis. The goal of this project is to develop a microarray-based diagnostic platform that can use both label-free and fluorescent imaging. Now, if you don't understand any of these terms just yet, don't worry. Hopefully, by the end of our presentation, you not only understand these technologies, but what we've done to integrate them. First, we'd like to start with a little bit of background on these technologies. To make a diagnosis, we are using DNA microarrays. A DNA microarray is a substrate with single-stranded DNA immobilized on the surface. These are called your probes. When a sample is washed over the substrate, your probes will capture any DNA sequences in your solution that are complementary to your probe sequence. These captured sequences are called your targets. And the binding of your targets to your probes is called hybridization. After hybridization, if you can then image your substrate and determine which probes have captured their target DNA, you can determine what sequences are in your solution. Now, it's important to note that also this process can be repeated with proteins, as the last demonstration showed. And instead, you would spot down proteins on your substrate and then incubate these arrays with their respective antibodies, and these same measurements can be taken. For this particular project, we have used a robotic spotter to print our DNA microarrays onto our substrate, which is just a machine that allows us to determine the exact location of each probe on the substrate.
Right. So our project focuses on 2 main methods of imaging, the first of which is called fluorescent imaging. Fluorescent imaging utilizes proteins that have the ability to emit light when scanned with a specific wavelength. These fluorescent tags are attached to your target sequences. Then, when your targets are bound to your microarrays, a laser can be used to scan the surface of the microarray, and the fluorescent tag will emit light. This way, you can make a detection of binding of either proteins or complementary DNA strands. A fluorescent image is pictured on the right side of the slide. The second imaging technique we're using is called label-free. It is called label-free because it does not require tags to be attached to your targets in order to image them. Currently, the method to do this relies on interference, which I'll explain shortly. But the important thing to note is that label-free measuring gives a quantitative measure, which will tell you both the height and the mass of the material on your substrate. So, without these tags, how does this label-free imaging work? The Unlu lab has developed a system called IRIS, which stands for Interferometric Reflections Imaging System, that uses interference to do this. First, your microarray is printed on a silicon dioxide chip. This is important because you can exploit the interference of the dioxide. First, the biomaterial is modeled with having the same refractive index as the silicon dioxide. Then a tunable laser is scanned over your biomaterial in your chip. As it scans through its wavelengths, it goes through constructive and destructive interference. Through these patterns, you can extrapolate both the height and the mass information of the biomaterial on your substrate. So now that you understand a little bit about fluorescent and label-free imaging, I'd like to talk to you about our task. Our task is to create a chip with an appropriate silicon dioxide thickness that will work with both label-free and fluorescent imaging. This will allow the user to get the sensitivity that fluorescence gives while getting the quantitative measurements from IRIS. So, in order to design this chip, we have to first understand the model of a fluorophore, which is the fluorescent tag I talked about before, sitting on top of your silicon dioxide chip. So here you can see the fluorophore lens. And so what happens is that you scan this fluorophore with a laser at the wavelength of your fluorescent tag. When the light comes in, it excites the fluorophore there, but it also reflects, since the silicon is reflective, off the surface. Given that the silicon dioxide is chosen at the right thickness, constructively to give you more light at your fluorescence. Then your tag will emit light, again reflecting off the surface and constructively interfering, producing a fluorescent enhancement. Now, commonly microarrays are printed on glass, and the fact that we're using silicon allows us to get this reflection, whereas with glass, most of the light escapes through the back of the substrate. So we define the fluorescent enhancement to be the amount of light we get over that which a glass slide would get.
Now that you understand a little bit about the background of this project and the technologies involved, we want to break down the overall project flow for you visually. So we start by simulating different oxide heights and examining the fluorescent intensities received for each oxide height. This information allows us to choose our specific oxide height, and then we are able to fabricate our chips. These will be our substrate for our DNA microarrays. With these substrates made, we can then test our chips and make sure that they do produce the fluorescent enhancement we hope for, and that they are compatible with iris, our two main requirements. On the side, our BME team members have come up with a strategy to amplify and extract single-stranded DNA, and for this particular project, we've chosen influenza A-derived DNA. And with this concentration, we were able to then use the sample to make a label-free detection using the iris system that we have shown you before. Now, it's important to note that the iris system has never been used before to detect extracted and amplified DNA from a cell.
All right. So as Craig mentioned, the first step to choosing this oxide thickness is to run simulations. So this is the result of a simulation we ran in MATLAB, showing where we varied the oxide thickness on the x-axis and looked at the resulting intensity on the y-axis. The green and red curve here show the Psi3 and Psi5 fluorophore, which are two common fluorescent tags used, sitting on the silicon dioxide substrate, while the blue and the black curve at the bottom show the same two fluorophores on glass. You can immediately see that glass does not give nearly the same fluorescent intensity as the silicon dioxide. So now we ask, which oxide thickness do we choose to make these chips at? Well, from looking at the graph naively, you would say 100 nanometers. This seems to give a peak for both fluorophores, and this gives our best fluorescent intensity. However, the problem with this is that the iris system has been optimized for 500 nanometers, and the farther away you get from the 500 nanometer point, the worse performance you get from the iris system. So then we ask, why not use 500 nanometers since it's already being used for label-free imaging? Well, the problem with this is it sits right in the trough of our Psi5 fluorophore, so when we use this, we won't get any good fluorescent enhancement at all. So we chose to use 320 nanometers, which sits near the peak of both fluorophores when they're on silicon dioxide, and will be closer to the 500 nanometers used by iris.
Now that we've chosen our specific oxide height, we can then fabricate our chips. So we took a bare silicon wafer up to the clean room and thermally grew 320 nanometers of oxide on top. This wafer was then patterned lithographically, producing the image you see to the right. Afterwards, the wafer was diced and cleaned, producing our final chips that you see here. The quadrants in blue are our 320 nanometer silicon dioxide. Now, the overall pattern you see on the chip is just a function of the lithographic mask currently used by the UNU lab. With our chips manufactured, we now want to test the chips and see that they produce the fluorescent enhancement that we think that they do, and also test to see that they are compatible with iris. To do this, we decided to spot a range of protein concentrations on each quadrant of our chip. By incubating, afterwards, we incubated these proteins with their respective antibodies that are fluorescently labeled so that both label-free and fluorescent measurements can be taken of our substrates. By running this experiment on both our chip and a 100 nanometer chip, as well as a glass slide, we can compare the results of these to see that we have a fluorescent enhancement. And also by running this experiment on our chip as well as a 500 nanometer chip, we can compare the label-free measurements to make sure that our chip is compatible with iris. Before we go into the quantitative results of this experiment, we want to give you a mental image of what the fluorescent results look like. Here on the left, you can see a picture of our 100 nanometer chip, and you can tell that it is fluorescing much brighter than all of these chips, but especially the glass slide pictured at the right. In the center is our 320 nanometer chip, which is slightly dimmer than the 100 nanometer chip, but again, you can tell that it is much brighter than the glass slide. Overall, we measured that the 100 nanometer chip produces a 10 times average fluorescent enhancement over glass, while our chip produces a 9 times average enhancement over glass.
So now I'll show you the plots of these results. And first we look at the fluorescent results. And remember that the goal of fluorescence was show that we can get a good fluorescent enhancement over a glass slide. So on this graph you see our 6 different spotting concentrations on the x-axis. And on the y-axis you see the fluorescent signal that we measured. The light blue bar on the left represents the glass slide. The red bar represents our 320 nanometer silicon dioxide chip. And the blue bar represents the 100 nanometer chip, which we found was optimal, although it didn't work with Iris. And as you can see, for each concentration, our chip gets a significant increase in fluorescence over the glass slide. And we measured that on average across all concentrations, this was about a 9 times enhancement. So now we look at the label-free results, and this is when we're using the Iris system to measure our 320 nanometer chip versus the 500 nanometer chip that the system was designed for. Again, what we were looking for is that our chip measures a mass change within 1 standard deviation of the mass change measured by the 500 nanometer chip. And again, for every concentration, you can see that our chip measures the same, or measures within 1 standard deviation of the same mass change. This means that our chip can be used to detect binding as a microarray and is a suitable platform for both label-free and fluorescent imaging. So now we turn to the next aspect of our project, which is to show that we can use label-free imaging to image an influenza sample. And so far in the lab, only purchased DNA has been seen, not anything that has actually been extracted and amplified from an actual sample. But in order to do this first, we need a sample. And to get that, we measured that we need approximately 200 microliters of 100 nanomolar concentration DNA solution. And we go to our BMEs to get this.
We start with influenza A virions and lysine. The solution is then run through solid phase extraction columns under pressure. These extraction columns are fabricated by embedding a porous polymer monolith that is designed in the Clapbridge lab in pipette tips. The RNA is stored in the monolith and can be later eluted with water for subsequent use. Here's a close-up of the columns. If you focus your attention to the rightmost columns, you can see this process in action. The RNA is being stored in the white monolith, while the waste, that includes other nucleic acids and proteins, drips down the bottom.
Now that we've extracted the RNA, we need to modify it in 2 ways so that it can successfully bind to these single-stranded DNA probes on the DNA microarray. First, the RNA influenza sample needs to be changed into a DNA sample. And second, that double-stranded DNA sample needs to be changed into a single-stranded DNA sample. So first, we reverse transcribed or made the RNA into DNA and amplified the sample using an RT-PCR technique. And what this does is makes many copies of this double-stranded sequence. We then attached a biotin molecule to only 1 strand of this double-stranded DNA sequence. We did this by using biotin-labeled primers during the PCR reaction. Next, we mixed the biotin-labeled DNA in solution with streptavidin-encoded magnetic beads, thus allowing the DNA to bind to the beads using the streptavidin-biotin bond. After binding, we denatured the double-stranded DNA in an NaOH solution and applied a magnet, thus allowing us to immobilize the biotin-related strand and then isolate the non-biotin-related strand. And this strand is what we then used to bind to the DNA microarrays. So in conclusion of sort of the BME side of the project, we were able to, using this developed technique, obtain 200 microliter solutions of 100 nanomolar single-stranded DNA.
With this solution now available to us, we want to again prove that we can use Iris to make a label-free detection of influenza A, a key portion of our overall project. To do this, we spotted the following configuration you see. This is composed of 2 different probes. One is our influenza A probe. This is a 20 base long single-stranded DNA, which is complementary to a portion of the influenza A sequence of interest. Then we spotted a control probe, which is a 23 base long single-stranded DNA that has little affinity to the influenza A strand of interest. After hybridizing this microarray with our solution, we hope to show that the influenza A probes change significantly in mass, while the control probes do not change significantly in mass, indicating that we have indeed correctly captured our influenza A sequence. Just to give you an idea of the procedure used to run this experiment, we first spotted this configuration onto our chips and measured the initial masses using iris of each spot. Then the chips were covered with the hybridization chamber you see here pictured to the right, and the overall configuration was heated up along with the sample to 63 degrees Celsius. This temperature was chosen because it is 10 degrees below the melting point of our target DNA, the influenza A DNA, which allows us to safely assume that our DNA is not forming any secondary structures, meaning it's not binding to itself in solution. After all of the pieces were heated to the correct temperature, the samples were pipetted into the hybridization chamber, and the chamber was then sealed off, and the overall configuration was allowed to cool slowly to room temperature for 1 hour. Finally, the substrate was washed, and the post-hybridization masses were measured with iris. Here's a graph depicting the results of this experiment. On the left is a red bar indicating the overall average mass change of our control probes, and on the right is a blue bar indicating the overall mass change of our influenza A probes. As you can see, the overall mass change for the control probes is about 0.1 nanograms per millimeter squared, while the influenza A probes have changed by about 7 times that amount. Also, it's important to note that by comparing the control sample or the control probes to the influenza A probes, we can safely say that the binding that has occurred is specific, meaning that we have definitely captured our influenza A target. Thus, we can successfully use iris to make label-free detections of influenza A. Just to summarize what we have done this semester, we have been able to use iris to make these label-free detections. We have developed a silicon dioxide chip that allows for both fluorescence and label-free imaging. And our BME team members have come up with a single-stranded DNA amplification protocol that supplies us with the solution necessary to make these diagnoses. In the future, we hope that all of these components can be integrated into a single lab-on-a-chip platform, and that possibly this lab-on-a-chip can be used in low-resource areas for global health applications. We'd like to thank Professors Unlu, Professor Kabodi, and Professor Clapridge, as well as Sunmin An, who has helped us throughout this project, and the entire Unlu lab, who's been there since we knew very little about biology when we started this project. Thank you. Any questions?
Yeah, how do you calculate the mass change?
So there are some published results by the Unlu lab that show there are simple correlation factors between height change measured by iris and mass changes. And these factors differ depending on what you're measuring, of course. So for single-stranded DNA, 1 nanometer height change is equivalent to about a 0.8 nanogram per millimeter squared mass change. So that's the calculation we use.
I read somewhere that a DNA molecule has about 200 billion atoms in it. How come it sticks to your surface at the very end of the molecule like that?
Right, so we didn't get into that during our presentation, but the way that we attach our biomolecules to the surface is we first coat the surface with a polymer that has free amine groups, and then we order our probe sequences such that they have an amine group at one end. And then when you spot the DNA onto the polymer, you let it go for about 12 hours and allow these binds to be made between the amine group on your single-stranded DNA and the polymer. It's great.
How does the concentration of your test solution compare to the concentration you'd have in a patient? And can you extrapolate anything about the efficacy of your test in terms of false alarm?
Well, first to address the concentration issue, the samples we were actually using were, we were using cell-free supernatant of lab-strain cultured MDCK cells, which are canine kidney cells, that we had then infected with influenza A virions. And the amount of influenza A that was cultured in these cells was very equivalent to what an infected patient having, who has the flu would have in them. So that correlated well to when we extracted, using these solid phase extraction columns, to what it would actually be if we were using a human sample. And on top of that, because we did use the polymerase chain reaction, or PCR, to amplify, the way this works is you run many cycles of PCR and you get a doubling of however much sample you have. So we ran 45 cycles of PCR, meaning that we amplified our sample by 2 to the 45th power, meaning that you can start with a very, very small sample and through this amplification process, always be able to get up to the necessary concentration to bind to the microarray. And then, sorry, I forgot what the second half of your question was.
Well, how effective you think this would be as a diagnostic tool, whether it would capture a certain correct diagnosis and a certain false alarm rate?
Okay, we didn't, for the purpose of this project, go into the statistical analysis of how to go about determining if there were false positives or anything like that, because there were some certain aspects of the diagnostic tool we didn't get into in terms of maybe amplifying many different sequences of the influenza A sequence so that you could be very sure that, well, if these 5 different regions of the genetic code bound to the microarray, then you can be very, very sure, which is generally what's used with DNA microarray experiments. And we also didn't get to a stage where we mixed a control probe in our target sequence, so you can tell how that will affect nonspecific binding, because with a real human sample, you'll have other sort of junk in your sample. It won't just be pure single-stranded DNA of your appropriate target sequence. So because we didn't get a chance to do some of these further testing that would be done in the future, we can't really determine the efficacy of how specific this diagnostic tool is at the present moment.
You showed in your analysis that with effective binding, you saw a fairly substantial change in mass. Was that with a sample of 1, or are your results statistically significant over a variety of different tests?
Sure. Well, so we said from, sorry about this plot. So we looked over, so we spotted this configuration that we showed right here on 4 quadrants of a chip, and you see there's 25 samples of our influenza probes, and there are 10 control probes. So we looked at the results from all these plots, and we took measurements, and we looked at the standard deviation, and you can see from our error bars that these results are significant, and that our control probes do have significantly less binding than our influenza probes. So yes, there.
What is the mass before you do this binding process?
So it depends on what you're spotting onto the surface. When you take your initial mass measurements, I found that when I spotted DNA onto the surface, measured about a 1 nanometer height change, maybe a little bit lower, 0.8. So multiply this by 0.8 to get your nanograms per millimeter squared.
Thank you.